Good morning. In part 1 video, we were discussing about the pathway of glycogen metabolism, both glycogen synthesis as well as glycogen breakdown. In this video, we will see how these pathways are regulated. Regulation is a process by which the metabolic pathways are controlled as per the body's need. Whenever there is a demand, the pathway will be switched on. On the other hand, when we don't need it, it will be switched off. This control will be executed through the rate limiting enzymes of the metabolic pathways. So now coming to our glycogen metabolism, for glycogen synthesis, the rate limiting enzyme is glycogen synthase. And for glycogen breakdown, it is glycogen phosphorylase. So these are the two rate limiting enzymes in glycogen metabolism. So both these enzymes, they are regulated mainly by two major processes. One is covalent modifications and the other one is allosteric regulation. First we will start with covalent modification. Covalent modification is a process in which the enzyme will be regulated by attaching a molecule to the enzyme through covalent bond. The most commonly executed covalent modification for enzyme regulation is phosphorylation and dephosphorylation of the enzyme. That means attaching the phosphate group or removing the phosphate group. Some enzymes, when you attach a phosphate molecule, it will become active. Some enzymes, when you remove the phosphate molecule, it will become inactive. So first we should know whether the enzyme of our pathway is active in phosphorylated or dephosphorylated state. So to remember this, I can give you a very easy and simple clue. For any metabolic pathway, you can apply this. Okay. So any pathway which leads to glucose production, the enzymes of those pathway will be active in phosphorylated state. Okay, P for P, you can remember glucose producing pathways, their enzymes will be active in phosphorylated state. On the other hand, glucose decreasing pathways, they will be active, the enzymes will be active in dephosphorylated state. Okay, so this clue you can remember it and you can apply it for any metabolic pathway in the carbohydrate metabolism. Now coming to our glycogen metabolism, glycogen synthesis. Okay, it is a process in which glucose will be converted to glycogen. So the glucose level in the blood will start decreasing. Okay, so this glucose decreasing pathway will be active in dephosphorylated state. Whereas glycogen breakdown, so here what happens, glycogen will be broken down and it will be released as glucose in the blood. So the glucose is produced in this pathway, so it is active in phosphorylated state. Okay, so now we will see in detail how these enzymes are phosphorylated or dephosphorylated and how they are regulated as per the needs. We will discuss in detail. First, we will start with glycogen synthase enzyme. So this glycogen synthase, which is actually the rate limiting enzyme of glycogen synthesis, this should be activated during well-fed state. When the blood glucose level is high, the glycogen synthesis pathway should be activated. On the other hand, in the fasting state, when the blood glucose levels are low, this enzyme should be switched off. This pathway should be inhibited. Okay, so how this is done, we will see in detail. Let us assume that the person is in a completely fasting state. Okay, so when he is in the fasting state, we all know that within our body, the anti-insulin hormones like glucagon and epinephrine will be produced in excess. So this glucagon and epinephrine, they will go and bind to the receptors present in the target tissues. Okay, so here for this enzyme, the target tissues are nothing but liver and muscle. Okay, so glucagon actually it acts only on liver, whereas epinephrine it can act both on the liver and muscle cell. Okay, so in the target tissues, these hormones, they will go and bind to the membrane receptor, G protein coupled receptor, and they activate an enzyme called as adenyl cyclase. So an enzyme adenyl cyclase will be activated. Okay, so this adenyl cyclase, as the name says, it cyclases the adenosine triphosphate and converts it into cyclic AMP. Okay, so this is the first step happening within the cell. Okay, so now this cyclic AMP produced will act as a second messenger and they will be activating another enzyme called as protein kinase A. Okay, so kinase is an enzyme which actually leads to phosphorylation. Okay, so this protein kinase A, it phosphorylates our enzyme glycogen synthase. Okay, we all know that in the phosphorylated state, glycogen synthase will become inactive. So this enzyme will become inactive and the pathway will be switched off. Okay, so this is how during fasting state, the glucagon and epinephrine, they phosphorylate glycogen synthase and they inhibit the glycogen synthesis pathway. On the other hand, suppose you are in a well-fed state. At that time, glucagon epinephrine levels will decrease and there will be an increased production of insulin in our body. Okay, so this insulin, what they will do means they will activate two major enzymes. One is phosphodiesterase enzyme and the other one is protein phosphatase enzyme. So through this enzyme, how they act means first phosphodiesterase. This phosphodiesterase, they will destroy the cyclic AMP within the cell. So if cyclic AMP is not there, protein kinase will not be activated. So phosphorylation of the enzyme will be prevented. On the other hand, the insulin, we saw that it is activating the second enzyme, protein phosphatase. So this protein phosphatase, as the name says, it is just opposite to the action of protein kinase. Protein kinase, it phosphorylated the enzyme, whereas protein phosphatase, it will remove the phosphate from the enzyme and converts it into a dephosphorylated form. Okay, so now glycogen synthase, we all know that it is active in dephosphorylated state. So in the presence of insulin, when it gets dephosphorylated, it gets active and it initiates the glycogen synthesis process. So in the well-fed state, the excess glucose will be converted to glycogen and it will be stored in the liver and muscle. Okay, so this is how glycogen synthase is regulated by glucagon and epinephrine, anti-insulin and anti-insulin hormones through phosphorylation and dephosphorylation of the enzyme. Okay, next going to the glycogen phosphorylase enzyme. So how this acts means, so how this is regulated means same way. Okay, so we all know glycogen phosphorylase is a process, is an enzyme used for glycogen breakdown. Okay, so this glycogen breakdown, when it should happen? Ideally, it should happen during the fasting state. Okay, whereas during the well-fed state, it should not be happening. Okay, so in the fasting state, glycogen should be broken down and should be released as a blood glucose, whereas in well-fed state, this should not happen. So how this is regulated accordingly means, so first initially same way. Okay, so in the fasting state, what will happen? Glucagon and epinephrine will be produced in excess. So this glucagon and epinephrine will bind to the G protein coupled receptors in the target tissues and they activate an enzyme called as adenyl cyclase. So this adenyl cyclase will cyclase the ATP to form cyclic AMP. This cyclic AMP again it activates protein kinase A. Okay, so in the previous slide we discussed that this protein kinase A will directly activate the glycogen synthase enzyme, but here there is one more step. This protein kinase A initially activates phosphorylase kinase and that in turn will phosphorylase glycogen phosphorylase. Okay, so now like same way how glycogen synthase was phosphorylated, even glycogen phosphorylase will also be phosphorylated in fasting state under the influence of glucagon and epinephrine. But what is the difference means, under phosphorylated state, glycogen synthase will become inactive. So the pathway will be switched off. Whereas here, the glycogen phosphorylase under phosphorylated state, it will become active so that the pathway will be activated. Okay, so thus during fasting state, under the influence of glucagon and epinephrine, the glycogen phosphorylase, the rate limiting enzyme of glycogen breakdown, will be phosphorylated, activated, and the pathway will be switched on. On the other hand, whenever you are in a well-fed state, what will happen means same insulin will be produced. This insulin in turn will activate two different enzymes. One is phosphodiesterase and the other one is protein phosphatase. So these two, as we seen in the last slide, phosphodiesterase, they will destroy the cyclic AMP and prevent the phosphorylation of the enzyme. On the other hand, protein phosphatase, they will remove the phosphate from the enzyme and it causes dephosphorylation. Okay, so when dephosphorylated, glycogen phosphorylase will become inactive and its action will be stopped and the pathway will be switched off. Okay, so this is how glycogen synthase and glycogen phosphorylase is regulated as per our needs by covalent modification. To sum up, so what exactly happens means, so the first you should know the enzyme is active in which state. Okay, for a mention for regarding covalent modification, initially you should know the enzyme is active in which state, then only you can proceed with the further things. Okay, so our glycogen synthase, it is active in dephosphorylated state. So once you know that, easily you can find out which hormone will activate it and which hormone will inhibit it. Okay, that is fixed. Insulin always causes dephosphorylation, whereas the anti-insulin hormones like glucagon and epinephrine will cause phosphorylation. Okay, so here since it is active in dephosphorylated state, insulin will activate it, whereas glucagon and epinephrine will inhibit it. Okay, so this is how glycogen synthase is regulated. Next, coming to glycogen phosphorylase, it is just the reverse. It is active in phosphorylated state. So the hormones glucagon and epinephrine will activate glycogen phosphorylase, whereas insulin, which usually causes dephosphorylation, will inhibit the action and it will inhibit the pathway. So this is how glycogen synthesis is regulated as per the needs during fail-fed state and starvation under the control of insulin and glucagon. Okay, so apart from insulin and glucagon, there are also a few other hormones which can also regulate the glycogen metabolism. They are nothing but vasopressin, oxytocin and angiotensin 2. So these hormones, they mainly regulate the metabolism within the muscle cells, especially within the smooth muscle cells. It will be regulating the glycogen metabolism. So how it regulates means their action is almost similar to the anti-insulin hormones like glucagon and epinephrine. That means they will phosphorylate the enzyme. So if it phosphorylates the enzyme, what will be its effect on the glycogen metabolism? Glycogen synthase will be inhibited, whereas glycogen breakdown will be activated. That is the net effect of these hormones. But their mechanism of action is quite different from the anti-insulin hormones. Okay, so anti-insulin hormones, how they phosphorylate the enzyme? So they activated adenyl cyclase, which in turn activated cyclic AMP. That cyclic AMP activated protein kinase A, and that protein kinase A led to phosphorylation. Okay, but here what is the thing is, here also the hormones will bind to the same G protein coupled receptors, but here the effector enzyme is different. Instead of adenyl cyclase, it will activate an enzyme called as phospholipase C. This phospholipase C in turn will hydrolyze phosphatidyl ionocetal bisphosphate to form ionocetal triphosphate and diacyl glycerol. This ionocetal triphosphate in turn will increase the cytosolic calcium level, and this cytosolic calcium in turn will activate another protein kinase called as protein kinase C. Okay, and this protein kinase C cause the phosphorylation of the enzyme and thus the regulation of the pathway. Okay, so that's it about the covalent modification of our enzymes involved in glycogen metabolism. Now coming to the second part, allosteric regulation. Okay, so this is actually very simple. So allosteric in covalent modification, the controlling element, it came from outside the cell in the form of hormones. Okay, it was glucagon or epinephrine or insulin or vasopressin. It was produced outside the cell, and they came and executed their action in the target cell. Okay, but whereas allosteric regulation, here the regulators will be within the cell. Okay, it will be usually within the intermediates of the pathway. Some of the substrates and the products involved in the pathway will be executing this regulation. Okay, so usually what will happen means the substrates, when they accumulate, they will bind to the enzyme and they will activate the pathway. On the other hand, when the products accumulate, they will bind to the enzyme and they will inhibit its action and it inhibits its pathway. Okay, so now we will see, first we will start with the glycogen phosphorylase enzyme. So for glycogen phosphorylase, what is the end product? So when you break glycogen phosphorylase, by the action of glycogen phosphorylase, glycogen will be broken down and it will be released as glucose 1 phosphate. That glucose 1 phosphate will be converted to glucose 6 phosphate. That glucose 6 phosphate will be converted to glucose. That glucose in turn will be utilized by the cells for ATP synthesis. Okay, so these are the products we get from the glycogen phosphorylase enzyme. So all these products, when they are in excess, okay, when they are in excess, what does it mean? We don't need the glycogen breakdown pathway. So they will go and bind to the enzyme and they will switch off that enzyme. Okay, so this is called as allosteric regulation. Okay, so whenever glucose 6-phosphate is excess, or glucose is in excess, or ATP is in excess, body doesn't need glycogen breakdown. So it will be switched off by these intermediates. Okay, on the other hand, the substrates, when there is need of energy, okay, when AMP is more, or when there is a need of energy, or whenever there is a need of glucose, then this pathway will be activated. Got it? Fine. Next, coming to glycogen synthase, it is just the reverse. Okay, we call it as reciprocal regulation. Okay, so whatever activators for glycogen phosphorylase, they will act as a inhibitors for glycogen synthase, and whatever the inhibitors will act as the activators. Okay, so this is how glycogen synthase and glycogen phosphorylase enzyme is regulated by allosteric regulation. Okay, so that's it about the regulation part of glycogen metabolism. So both the enzymes, they are regulated by two different processes. One is covalent modification and the other one is allosteric regulation. So in covalent modification, the signal comes from outside the cell in the form of hormones. Okay, so hormones, insulin will cause dephosphorylation of the enzyme, whereas anti-insulin hormones will cause phosphorylation of the enzymes. Okay, this is fixed. Okay, but some enzymes, it will be active in phosphorylated state, and some enzymes will be active in dephosphorylated state. So accordingly, the net result differs. The effect, final effect differs. Okay, so glycogen synthase is active in phosphorylated state. So glycogen synthase in dephosphorylated state. So insulin, which causes dephosphorylation, will activate glycogen synthase. On the other hand, glycogen breakdown, phosphorylase enzyme is active in phosphorylated state. So the anti-insulin hormones will activate it. Okay. Next, coming to the allosteric modification, it is very simple. The products will inhibit the enzyme, whereas the substrates will activate the enzyme. Okay. Thank you.